while we get organized. I'd like to welcome on stage Elijah. Am I spelling it correctly? Am I saying? Elijah Willie, who is going to tell us about FuseSOM, a new correlation-based clustering algorithm for in-situ multiplexed cytometry imaging. Please join me in welcoming Elijah on stage.
Okay, is that good? Okay, hi everybody. Thank you for having me. So my name is Elijah. I'm a first year PhD student at the University of Sydney. And today I'll be talking about my first year project, which is a clustering method for imaging mass cytometry data. So by way of introduction, as some of you may be aware, imaging mass cytometry has really enabled us to look at cell with very high resolution. We're now able to capture the expression of multiple cells within the image, which give us some spatial locations. We can then do analyses with it. This is a very powerful tool. And that's with that, defining cell types is sort of a gateway to all of our downstream analysis. If you're doing single cell technology, it might be interested in differential expression. So before you do that, you need to sort of figure out where your cells are and how they sort of combine with each other. So clustering is a key for that. And there have been many methods developed for single cell, but not so much for cytometry data. But there are a couple that are used, for example, FuseSOM and Phonograph. I'm sure some of you here might have used those before. And as with clustering, you typically define distances between points and then use that to sort of aggregate them. And one of the most common ones is Euclidean distance. So there was a paper that was sort of came out a few years ago that sort of looked at the impact of these distances on how well you can cluster data. And it showed that for single cell, using correlation-based matrices like Pearson's pyramid does actually outperform the typical Euclidean. So we were interested in trying to figure out if this is the same for imaging data. And as with single cell technologies, there are problems that are inherent to imaging mass cytometry. So for example, on this picture here, you can see there is some bright spots, which you can try to normalize out. You can get rid of some of it, but at the end of the day, these artifacts are still part of your analyses. And when you use distances, you have to be careful because, for example, using Euclidean might weight these bright spots the same as every other cell, whereas some other distances might not. So we're trying to figure out a way how this can be assessed for this kind of data. And so the aim of this project was to look at the efficacy of correlation-based matrices on these and sort of combine these ideas into a package that's freely available for people to use. So by way of example, this is a data set that was published recently on lung tuberculosis. And so here I have included the UMAP plot of the cells and their annotation. So these cells were clustered and then manually annotated using expression data. So there is some level of difficulty in recapturing these cell types. I then picked a random algorithm, hierarchical clustering using correlation and Euclidean. I chose Pearson. And you can see that for the data set cluster using Pearson, we have a performance that's double that of Euclidean. And the values here are quite low because, again, this was manually annotated after clustering. So we're not just matching the output. And I used the adjusted random index, which is a metric that tells you how well two sets are how well they match. So a value of 1 means you perfect match, and value of 0 means you don't match so well. So to see if this idea was consistent across multiple data sets from different technologies, we included a few more data sets that are ranging from SciTOF, MeBTOF, and also SeqFish to see if we could sort of recapture this. And so these data sets were clustered using the same algorithm with all these distances here, and their values were ranked with respect to their average random index. And here a value of 6 means it's the best and value of 1 means the worst. You can see that at the bottom here, for most of the data sets, the Pearson, cosine, experiment are always in the top. So this is just consistent for one data set, is consistent across quite a few data set. And again, we wanted to know if this was just maybe an artifact of one clustering method. So we performed this across a few more, including ones that typically use, for example, FuseSOM. And so this now is average across all of the data sets I showed earlier. You can see that for a lot of the methods, the correlation metrics does outperform the Euclidean one, except for phenograph here. But even if you consider phenograph, the average performance for the other methods still does much better, regardless of using correlation or Euclidean for phenograph. So we've established now that across clustering algorithms and across data sets, correlation seems to perform quite well. The next idea was to see if we could sort of combine these distances into one, sort of maximize the signal from each one of these metrics. So we decided to pick 4, Pearson, Spearman, cosine, Euclidean, and sort of generated a Cartesian product of all of them and use that to cluster data set. You can see in this plot, again, for the ARI, for the times where we do combine more than one distance, we do get the most optimal performance. So there is something happening there where we can sort of combine these metrics into one and get that. So the next final step was to sort of combine these ideas into one that you can sort of use. But with that, with imaging data, you have data sets that are ranged up to 1 million cells, and computing correlations with those ones are almost computationally, it doesn't make sense to do that. So we had to reduce the data set in some way to maximize these guys. So we chose to use the Self Organising Map, so which is a similar technology that's used by FuseSOM. So this works by taking your data matrix and then finding prototypes where each prototype represents a set of points. So in this case, you can see you have 3 points that will map to this one, and then so on and so forth. These prototypes are then computed, distances are computed on these prototypes, Pearson, correlation, cosine, Euclidean, experiment, and then those distances are then merged and then clustered here. The clusters then map back to the original point. So this reduces the computational burden by quite a bit and is able to keep the speed and also efficiency of the algorithm. The next step was to then compare this method we called FuseSOM to what's typically used nowadays by practitioners, FuseSOM, across a bunch of imaging data sets from different technologies. And so FuseSOM was run just natively as you would a bioconductor or CRAN with other the default parameters. And you can see that for almost all of the data sets, FuseSOM outperforms in terms of ARI. So this tells me that we're obviously doing something right, and it's showing in these plots. The next step was to sort of extend FuseSOM further, because when you cluster data, you need to have an idea of how many clusters you have, and that's more of an art than science. So we implemented some well-known methods for cluster estimation. This here is just a relative error, which gives you the difference between your predicted number of clusters and the true number of clusters. So we can see that here the jump and the discriminant method are the ones that sort of have the best performance. But this is just a suggestion. You can sort of overcluster, undercluster, and do whatever you like. And just to follow up with that, if you do decide to go with those automated methods, the performance are not so bad across the data set. It's similar to when you just pick the number of clusters from the published papers, try to recreate whatever the dates. They jump here and the discriminant are again the top performers. So I'm a computer scientist by training, and I'm interested in seeing computational complexity. So I was curious to see how long it would take me to run about 1 million cells or 800,000 cells in FuseSOM, and sort of how much memory will I use. So FuseSOM, in terms of the number of cells, is a linear algorithm. So if you were to run I ran this on my computer for 800,000 cells, and it took about 8 GB memory in the maximum. And then it took under 5 minutes to run that from start to finish. So it's fast. and it's quite efficient. So just to conclude, we've shown that correlation-based matrices does do well for clustering data, and especially so with this kind of data. And by integrating multiple distances, you can actually improve performance. So FuseSOM has then integrated all these ideas into a package that's easily accessible, and it's been accepted onto Bioconductor, and it's available now for you to try. So all of this work couldn't be done without my supervisors and other people in my lab. So like I say, let's give to Caesar what it is. And I'd like to thank everybody who contributed this in one way or another, and also the Sydney Bioinformatics Center for providing a research conditions for me that made me thrive, and also my funding from my university. Okay, that's it. Thanks, guys. I'll take your questions.
Thank you, Elijah. Are there, oh yeah, Marie.
Thank you, very nice talk. I was wondering, could you give a bit of explanation? The correlation is being computed at the single cell level or on the meta clusters?
So the correlation, I think it's sort of n squared in number of points. So if we have a 800,000 by 800,000 thing, you would get like a huge matrix. So it's computational. It's too high of a computational burden to do. So I just computed on the meta cells or the prototypes. Yeah.
And then I was wondering, how is that affected by the number of clusters you are selecting?
The number of clusters I'm selecting. How is what affected? Sorry.
I mean the different measurement, the distances.
So the number of clusters I'm selecting is sort of post hoc to that. So I compute the distances on the prototype. So for example, if I were to project a 800,000 by 20 matrix to a 10 by 10 matrix, so 100 points, I would then compute these distances on that and then cluster the distances and then project, because I can map it back to the original cells. That's where the speed comes in.
Okay, thank you.
We have one more here.
Hi, Eliza. Very nice work. Great to see FuseSOM being used in this manner. I've used FuseSOM a lot in the past and find it quite surprising how the ARI was so low. Did you explore different grid size number of meta clusters? Yeah.
So I didn't put this in my talk, but my method actually automatically detects the grid size as optimal for clustering. So FuseSOM, I think, uses a 10 by 10 grid. So you generate 100 meta cells or meta clusters, and then you consider that my just looks at the distribution of sort of your eigenvalues of your covariance matrix and just figure out what's the best place to cut it off. But I also provides you the chance to supply your own. So you can either use the data to figure out what your grid size should be, or you can say, I would like to try something. So it leaves room for exploration. I just made it easy that you can just hit run, and then everything is done for you.
Yeah, okay. Thanks for that.
Thank you. I think we might go with our next speaker, but please join me in thanking Elijah again.